Okay. Well, welcome everyone. Thanks for coming to the last colloquium of the semester, which I think would be exciting end to this season. I'm delighted to introduce Michael Desai, who is at Harvard University now. He is appropriately the Fisher Professor of Natural History across sort of organismal and evolutionary biology and physics. And you'll see how I think he straddles the two very, very well. Michael did his bachelor's or PhD at Harvard, and then spent a postdoc as a Lewis Sigler Fellow at Princeton, and then is back in Harvard now. And notably, he was the Simons investigator in modeling and life sciences and things like that. So, well, Michael.
All right, thanks, Sid. So first of all, please feel free to interrupt and ask questions at any point. The talk is kind of modular. We can get through more or less of it, depending on how many questions there are. That's all fine. And this is sort of a new area for my lab, looking at immune viral coevolution. I'm certainly not an immunologist, arguably not even a biologist, but I will do my best to answer your questions and tell you about what I think is a really, really cool system and set of questions. So to begin with, I just want to take a minute to remind you, you all know quite a bit actually about immune viral coevolution, even if you have never really thought about it from a scientific point of view. So you all remember in 2020, something bad happened. Okay, in 2025, something else bad happened, but that's not immune viral coevolution. Okay, so in 2020, you all remember COVID came up. And for a solid year or so, we just had COVID, okay? We didn't have all these different strains of COVID. COVID was like basically not evolving, okay? And that's basically, we now sort of understand, because COVID was entering a previously immunologically completely naive population. None of us had any pre-existing immunity to COVID. But during this period, we were starting to build it up. And then in early 2021, you all remember, the vaccines came out and everybody started getting vaccinated. And now all of a sudden we started to develop population-wide immunity to this original Wuhan strain of COVID. And then you may remember Alpha came out primarily in England and then spread across the world. And then Delta came out and then Omicron. And there's been this constant turnover since then. And as these strains have come out, you know, they've escaped pre-existing immunity, and then correspondingly, both through infecting people and through additional updates to the vaccine, have had to sort of basically adapt to evade the pre-existing immunity that the previous strains had themselves created. This population immunity we can think of as, you know, initially it was non-existent, then individuals started acquiring immunity and antibodies that effectively neutralize these earlier strains. As new strains come, sort of with some amount of time lag, there's increasing immunity against an increasing breadth of previous strains, but there's a constant sort of arms race going on. The virus is always evolving forward to evade all of the pre-existing immunity. Now, there's a huge amount that we can say about all this process, and the way in which the immune system works is enormously complicated, and we're not going to really talk about most of it. As I said, I'm certainly not an immunologist, but the coevolutionary dynamics really depend on molecular interactions binding typically between these antibodies and viral antigens. So in the case of COVID, this is primarily the spike protein, which is one of the surface proteins, which is a primary antigenic target. In influenza, which I'm also going to talk about, it's hemagglutinin. And really all of this coevolution is based on the complex sort of nature of molecular interactions between the antibodies and the virus, which, you know, the virus is constantly evolving to sort of eliminate those interactions, and then our immune systems are evolving to keep up. like I said, there's a few different things I'm going to talk about. I'm not sure how far I'm going to get, but we're going to talk about two general systems as examples of this process and how we can understand it. We'll talk about anti-influenza BNAbs. This means broadly neutralizing antibodies. So we'll talk quite a bit about this in a few minutes, but these are antibodies that protect you against different strains of influenza. And then, so that will sort of be a focus on evolution on the immune side of antibodies that protect you against a breadth of different viral strains. And then on the flip side, we'll talk about the evolution of a viral protein, specifically the SARS-CoV-2 spike protein. And we'll talk specifically about the evolution that led to the emergence of the Omicron variant, which was one of the first, you probably remember, one of the first sort of major immune escape variants that basically infected people regardless of any previous immunity to earlier strains. Okay. So we're going to start by talking about this anti-influenza BNAb. So this is the structure of the heavy chain of an anti-influenza antibody. Sorry, this here, the heavy chain here and the light chain. So this is part of the antibody. This is hemagglutinin, influenza hemagglutinin, which is one of the primary proteins on the surface of hemagglutinin. It's very important for the virus because it's what binds to the host cell receptors and gets into your cells. Kind of like the spike protein is what binds to ACE2 on the surface of your cells and gets into and infects you. And it's also because it's sort of sticking up at the surface of the virus is the primary antigenic target, which the immune system tends to attack. Okay, and well, here let me talk about how this works. I'll come back to this structure in a minute. Okay. So what happens when you get infected or vaccinated against some virus? You guys all probably have how many of you guys have had like COVID vaccines, influenza vaccines? So you all have personal experience. You have the COVID vaccine. How many of you felt really nasty for a couple of days? Okay. All right. Better than average, actually. Okay. That feeling nasty is because of an innate immune response. You get, infected or you get vaccinated. First, your innate immune system responds, makes you feel crummy. It's, causing all kind of inflammation. All kind of stuff is happening. There's a variety of different players involved. But what we're going to talk about is what happens. That innate immune system basically grabs some of the virus and pulls it through your lymphatic system to your lymph nodes. Many of you have noticed you get vaccinated or infected, the lymph nodes swell up because the stuff is going there. And there's a whole complicated set of things that are happening in the lymph nodes. But you've got these sort of proto-antibodies floating around in your blood, okay, in these things called B cells, okay, which is primarily what we're going to talk about. Each of these B cells is constructed via a kind of random process of recombination. So, you've got a finite genome. How many of you know how this works? Sorry, I get a sense of the audience a little bit. Okay, so most of you didn't raise your hand. So I'll explain the 50,000-foot view. You generate like 1 billion or more different B cells that express different like proto-naive antibody sequences, okay? And the way you generate that enormous diversity is through a combinatorial process. There's like a bunch of different V parts, D parts, and J parts that sort of combine together randomly to make this proto-antibody, okay? And then you've got tons of these floating around in your blood. And when you get vaccinated or infected, they kind of come into the lymph nodes, okay, together with the innate immune system bringing the pathogen into the lymph nodes. They form these structures called germinal centers. And basically, you're testing these naive B cells against this antigen, and you'll find one eventually that kind of binds kind of poorly. And then what happens is you start this, what's called an affinity maturation process, which is cycles of hypermutation and selection, which is evolving this antibody to bind better and better to that particular strain of that particular virus. And so what happens is you get this antigen, you select for binding. If you don't bind, you die. If you do bind, then depending on how well you bind and other things that are going on, not all of which are fully understood, you can go out into the blood and become a plasma cell and basically pump out antibodies that are going to now protect you from that virus. You become a memory cell, potentially, which will protect you in the future, many years later, against this same strain. Or you'll continue back and mutate further and try to generate even higher affinity and better antibodies. So it's like very much an evolutionary process that's sort of mutation and selection that's generating these improved antibodies over time. Okay. So that's like how the immune system evolves to protect you against a virus, okay? But as you all know, the virus is also evolving to evade this immune response, okay? So this is a sort of very, very broad view of diversity in influenza. So there are these two major groups of influenza A, and then there's influenza B, okay? And within each of these groups, like the currently circulating strains in humans are H1 and H3. These are the things you'll get vaccinated against every year if you get an influenza vaccine. And within these, there's also an enormous amount of diversity, and they're constantly evolving. And if you get vaccinated against an H1 strain that's circulating now, it'll generate immunity that, you know, a few years later, whatever influenza H1 is around then will have evaded that. Okay, so there's this constant sort of coevolutionary dynamics going on. What I'm going to talk about is why it's sort of hard for the antibodies to evolve to sort of keep up with this influenza evolution. And I'm going to talk about it by sort of looking at the opposite case, okay? Very usually you get an antibody when you get vaccinated that protects you against one particular strain of one particular type of these things that will be around for a very few years, okay? But occasionally, lucky people generate really rare antibodies that are called bNAbs, which will broadly neutralize lots of different stuff, okay? So I'm going to talk about two specific broadly neutralizing antibodies against influenza, CR9114, which basically neutralizes you against everything, and CR6261. This is the most broadly neutralizing anti-influenza antibody ever produced, and this is a broadly neutralizing, but not as broadly neutralizing antibody. This thing protects you against pretty much everything. This protects you against just these things. Okay, these were isolated through a whole sort of fishing process from some Dutch graduate student, you know, 15, 20 years ago. Okay, I met his PI, okay, who figured this out. If we could all generate this antibody, we would be immune from flu for life, and the flu, if we could all do it, would probably go extinct, okay? But we don't. So we're going to investigate like how this thing might have evolved, okay? Of course, there's a huge amount of interest among immunologists in these kinds of broadly neutralizing antibodies. They come up rarely also like in HIV, for example. There are like super neutralizers who generate like broadly neutralizing anti-HIV antibodies, and you know, they can live happily with HIV. But okay, this is influenza. Typically, this head of the influenza, which is kind of sticking out, is where the antibodies bind, and this evolves rapidly, but occasionally these antibodies evolve that bind to this stem, and these two are examples of that, which is more conserved because it's kind of sitting in the HIV sort of structure, okay? And so people have, you know, this is a co-crystal structure of HA with this antibody and with this antibody, okay? And, you know, these are, quite a bit has been sort of described about how they bind, okay? But there's less understood about like how did they evolve, okay? And what we're going to talk about is how we tried to map the sequence affinity landscape that characterized how this antibody evolved, okay? So what we're going to do is we're going to start from what we call the germline sequence. So that's like at the point where it was that naive antibody floating around in your blood. It had been generated by some random recombination. Okay, so that part, it's like not so rare. We probably all have something floating around vaguely like this naive antibody in our blood. And then in that affinity maturation process, in this Dutch dude, okay, it accumulated some mutations that generated this remarkable antibody that we would all love to have, okay? And what we're going to do, we know what the germline sequences are, possible germline sequences are. So we have the evolved antibody sequence. We infer the germline. There's a bunch of mutations. Okay, and we're going to sort of, they're more than this, but basically we're going to look at all the combinations and try to understand the structure of this landscape which generated this antibody. Okay, that's what we're going to do. And, you know, we're going to ask, like, is it sort of a smooth landscape or is it some complicated? Presumably it's complicated, otherwise we'd all find this antibody. All right. So this is the sequence. At the top, this is 9114 here. This is 6261. This is the germline amino acid sequence of this antibody, the heavy chain of this antibody. These are heavy chain binding antibodies for those of you that care about such things. Okay, and there are 16 mutations that arose in this antibody during this affinity maturation process. And in this antibody, I believe there were 11. Okay. So basically, we wanted to construct all combinations of these 16 and all combinations of these 11. In work that we're doing, actually, like we were working on this yesterday in the lab, these germline sequences are also quite similar. They're almost identical, with the exception of a little bit here. We also want to generate libraries involving combinations of these things to understand why this thing became broad but not as broad as this. And like, are there like contingency or branching points in the evolution? But that I don't have data yet. So here we're just going to look at these two individually. The way we construct these, there's like tricks that you can do. But basically, you can construct, so there were 16 variants in this 9114, so we created all 2 to the 16 combinations. Okay, and we did through various tricks. You can just order 66 DNA fragments and allow them to combinatorially come together, and you can generate them. And then what we wanted to do was we wanted to ask, how do each of these different sequences bind different influenza variants? Like, and how did they acquire increased binding to a really broad range of different flu variants? Okay. So to do that, we take this library of antibodies, okay? And for those of you that know, like, we kind of have to make some sacrifices here. We're not like looking at the whole antibody. We're just looking at the heavy chain of the antibody expressed in what's called this SCFV format on the surface of yeast. So it's not really capturing the full complexity of the complete antibody, but given what we know about how the binding works, we think in this case it captures the critical aspects of that particular binding interface. So we put these in, okay, and we transform, and then we've got a set of a yeast population. Each yeast cell is expressing one of these antibody variants on the surface of its cell, and it's got a plasmid with that antibody sequence inside it. Okay. And then we label it with one of our hemagglutinin variants. This we do produce, like the hemagglutinin. And so we've got hemagglutinin with a fluorescent label on it floating around. And basically, if a particular antibody variant binds that strain of hemagglutinin, you'll see it on the surface of the yeast cell, and that yeast cell will be glowing. Okay? And the better it binds, the more it'll be bound, and the brighter it'll glow. So we take it down to a flow cytometer and we sort the cells into bins based on how bright they are. And then we sequence those bins to find out which sequences are bright and which are not. Yeah. Flow cytometry, it's like basically it's a combination of optics and fluidics. You take your population of yeast, okay? And basically what you do is you flow them through a chamber, one cell at a time. And it's really cool to watch. Okay, you flow these things through a chamber, one cell at a time. There are lasers that are pointing at them, right, and exciting the fluorophores if they're bound to these fluorescent proteins. Okay, and then there's a detector that detects how fluorescent that particular yeast cell is. Then that yeast cell goes down and it comes in the stream here, and we've got a sort of magnetic plates. And based on the fluorescence, you can sort of put gates, like if it fluoresces in this way, in these channels, excited by this laser, but not that laser or whatever, you can sort of shoot it with the magnet into one of several tubes, okay? And you can do this at like 5,000 cells a second. Okay, it's wild. Okay, so in a few seconds, I can like I've got a library of 65,000 of these things, and I want to see like how each of them is fluorescing, and I want to do like 100 cells each. So I'm doing like millions of cells in like minutes. It's really cool. Okay. And you do this, okay, and then you sequence each of those bins, okay, so you know which cells are fluorescing at what level. And you do this at a bunch of different concentrations of this antibody. So then you can see, okay, this particular strain, it's not fluorescent at these concentrations, but then it starts to bind at these, so you can infer a binding affinity for 65,000 things in parallel, okay, in like a day. Okay, so what we did here, we picked 3 representative influenza variants to do this with that represent type 1 and type 2 influenza A and then a flu B strain to sort of represent the breadth of this antibody. And here we picked 2 pretty different type 1 influenza A strains to represent the breadth of that particular antibody. Okay. We did this, we were just kind of messing around, okay? And I did not really have high expectations for this project. And then we got this slide, this result, okay? So what I'm showing you here, there are 65,000 dots, okay? 1 dot per antibody variant, right? And then these axes are the binding affinity to H1, H3, and flu B. Okay, so higher, it's like the minus log of the binding affinity. So higher is stronger binding. Okay, and this is the germline here. This is the evolved antibody sequence. Okay, yeah, what am I going to say? The color represents like how evolved the sequence is. So out of these 16 mutations, how many of those that variant has. And so what you can sort of see is that like initially you have like a little bit of binding to H1. You could certainly get worse. Okay, there's bad things you can do. You can get better. All right, but you have to get better at H1 before you can acquire any binding to H3 at all. Okay, and then you start acquiring binding to H3, and then you can start acquiring binding to flu B. So like this immediately suggests there's some kind of highly contingent kind of sequential exposure that's needed to get this remarkable antibody to evolve. Another thing you see is that the antibody that we have is actually not the best possible thing. So when we publish this stuff, when we put this stuff on bioRxiv, I got a call from the guy who discovered this antibody, and he had started a company. He was like trying to squirt antibodies up people's nose, and he says, I don't want to make this, I want to make this, and can we work together on this? So it's actually, I mean, maybe not surprising in retrospect, but kind of cool that that's the case. Okay. The other antibody looks very different, right? This guy, you start here, you wind up here, and you can just kind of steadily get better at both. So somehow this thing is evolving very differently. All right. Now, we have the binding affinity to all these antigens of every one of these 65,000 variants. So I can actually like map the landscape in more detail, okay, than I've just described. And we can sort of really kind of try to work out that high dimensional landscape that I was kind of drawing pictures of. It's hopeless, of course, to plot it on a screen. So what I'm going to try to show you instead is a sort of low dimensional visualization that I think sort of captures the thing what's going on. What we're going to do is make a force-directed graph. So each point is going to be an antibody sequence, one of these 65,000. We're going to do 9114 here because that's the coolest one. And each point is going to be an antibody sequence, okay? And I'm going to connect the sequences that are connected by a single point mutation. And then I'm going to set a weight as 1 over the delta log KD. So it's going to tend to pull together sequences that are genetically close together and that have similar binding affinities. Okay. All right. And this is what it looks like when I do that for all 65,000. here's the somatic, here's the germline. So a way to, and these are the binding affinities. The way to think about these things are close together. Like to make a big move, you have to have a strong effect mutation, right? If you're interested, there's like a whole interactive browser for this over here that a student of mine, Milo Johnson, made, which is really cool. I'm just going to show you, these are screenshots from that. These are mutations at 5 of the different residues in that antibody. So what I'm doing is I'm coloring each of the points by the genotype at those 5 sites. So there are 32 possible colors, okay? And what you can see is these 5 residues tell you what you're binding, where you are in this landscape. Because like if you make mutations at other residues, you stay in this cluster, but when you make the green mutation there, you move, right? So this is like, these are the ones that determine the landscape. And just to sort of convince you of this, this is just 5 random ones, right? So that's what most sets of 5 will look like, right? So we could look at these 5 specific mutations and sort of see there's a complicated sort of 5th order interaction that determines the binding here. There are 5 mutations that have to interact in specific ways to make this antibody work. Okay, and we found these 5 by like looking at this thing, and then we started to look at, okay, these are the 5 mutations in the co-crystal structure. This is the hemagglutinin here. This is the antibody here. You can see they're kind of right at this interface, okay? And what's happening? Turns out this is the binding affinity of the to each one, okay? The first thing it gets binding to, all right? Without any of these 5 mutations. And if you get this mutation here, this 30S, you get much worse. Okay, so this mutation is bad for binding from the antibody point of view. These mutations are neutral, and these mutations are beneficial. But look what happens if I have this mutation. Sorry, what do I mean to say? If I have this mutation and now I make this one, okay, it's no longer deleterious. Okay, and if I have these 2 mutations, and I make this mutation, it actually becomes strongly beneficial. Okay. So what's happening is essentially this thing wants to create a new contact, but it needs to sort of reorient the structure of this protein at the binding interface to make it possible. Okay, and so you get this sort of complicated interaction that all sort of has to work together for this to acquire immunity, acquire this binding to this H1. That gets you binding to H1, all right? But then we can look at the binding to H3, which is the type 2 influenza A, and only a small fraction of the landscape has any binding at all. And all of those have 3 of these 5 mutations, okay? So you kind of have to get a little ways along this path involving this complex epistasis before you can even start to acquire broader binding to more diverse strains, okay? And then to get binding to flu B, you have to get even more, okay? So the implication here is that what happens and what causes this thing to be rare is that we all have this germline or something like it floating around in our blood, okay? And we all get infected by H1 influenza all the time, okay? It happened to me about 6 weeks ago, okay? And normally we create immunity to to that infection via some simple mutations in the antibodies that cause it to bind the head. But every once in a while, very rarely, we might get this sort of complex set of mutations that work together to create binding to the stem, okay? And then if and only if we got binding to H1 via this sort of unusual and unlikely complicated path, then we are in a point at which we can begin to acquire immunity to something broader, to H3. And we would have to then have gotten lucky to get this antibody in this way, and then again got infected by H3 to get it to get these additional mutations. And then I'm not going to tell you the full story, but then you have to acquire immunity to H3 in a specific way involving 5 additional mutations before you can start acquiring even broader immunity to flu B, right? So it's this sort of case of like this, there are ways to sort of navigate these landscapes to these amazing antibodies that would cure influenza, but because of the sort of structure of how hard they are to find versus easier things, we typically don't do it. And that's what, you know, makes this sort of, then we have to constantly update and keep evolving additional antibody variants. This is just a summary of that. Okay. There haven't been that many questions, and I still haven't gone very fast, but that's all right. So I want to shift gears now to the opposite side, okay? Now I want to talk a little bit about the evolution of immune escape on the viral side, okay? So this project started around Christmas 2022, okay? And at that time, you may remember, well, right around Thanksgiving, everybody in the world had Delta. You all remember this? Everyone had Delta, okay? I was actually running I spent the pandemic building and running the surveillance testing lab for Harvard and MIT. We were running like tens of thousands of COVID tests a day. We like built it over 2020 from the summer through like winter, and then like the vaccine came out just as we were getting up and running, and we were like, oh man, we spent like all these millions of dollars building this lab, and now the vaccine and the pandemic's over, and then Delta hit, and you're like, yes, okay, all right. So that was that time, okay? So Delta hit, our lab was like humming, everybody had Delta. And then Omicron came out, okay? And, you know, I had all these sequences. Everybody had all these sequences at the time. People started sequencing it. And we were like, where the hell did this thing come from, right? Because everybody had Delta. It's not that we didn't expect there to be additional variation generated, you know, additional variants generated, but they should be like descendants of Delta. Because like Wuhan was like practically extinct at that time. But instead, what happened is Omicron came, and it was a descent. It didn't descend from Delta. It sort of descended from this strain, which nobody had anymore. So like, how the hell did that happen, and why did that happen? Okay. I don't have that much time, so I'll give you the very quick thing. This is the structure of the spike protein of COVID. It's like the sort of analogous to the hemagglutinin in influenza. It's the protein at the surface that binds ACE2, which is your receptor that like gets you into the cell. If it doesn't bind that ACE2, it won't get in. And also it's the primary, this RBD is the primary target of antibodies. So when you get infected, like most of the time, the antibodies you generate are going to bind here. Omicron had 15 mutations in this RBD region. So we thought 15, that's a nice number. Let's make those. So we did the same game. We made those 15, okay? And what we find is this is the binding to ACE2. You've got to bind to this if you're COVID, otherwise you don't infect. And what we find is this here, this here is the binding of the Wuhan, the ancestral. This is the binding of the Omicron. And you can see most of the things in between are bad. So like somehow this navigated some complicated landscape, which like most ways of getting these 15 mutations are going to involve losing binding to ACE2, which would mean this thing couldn't work as a virus, or at least not in humans, okay? And you can see as you accumulate mutations, you go down and then you go up, okay? That's not to say there isn't an uphill path. Like maybe there's some very specific combination that works, but in general, there's this valley. Okay, I'm going to sort of skip some of this because I want to tell you about this story, okay? Bottom line is, what we found is there's this sort of fascinating sort of compensatory epistasis that allowed this Omicron variant to be created. So what I'm doing here, these are the 15 mutations, okay? And these violin plots show the effect of that mutation on ACE2 binding, you know, the distribution of that across all the 2 to the 14 background genotypes at the other loci, okay? And then I'm highlighting 2 of those other genotypes at the other loci in blue and red respectively. This is all the other loci are the ancestor, and then I'm asking what the effect of making this mutation is. And the red is all the other loci are the evolved Omicron, and I'm saying what is the effect of this mutation. What you can see, the blue points are pretty much always below the red. And particularly in some of these cases, they're way below. This mutation, for instance, it really destroys host receptor binding if it occurs in the Wuhan background. But if everything else is Omicron-like, it's fine. Okay, so there's some sort of compensatory epistasis going on. Okay, and I'll tell you, sorry.
I have an online question that says, does this imply an ideal vaccination order to increase the chances of generating this BNAB?
Yes, exactly. Sorry, I thought that, yeah. I didn't say that explicitly, but that was hopefully implicit in what I was talking about, right? Going back to the question is about the influenza story. And there, yes, I mean, what that implies is that if somehow we could figure out a vaccination regimen that, you know, involved sequential exposure to H1, H3, flu B, maybe we could make these kinds of broadly neutralizing antibodies more likely. Exactly. That's the sort of implication. I mean, to actually do that, I don't know, but that's the implication. Yeah. Okay, back to this. These particular loci, okay, where the Wuhan variant, where making this mutation in the Wuhan variant is really bad, but making it in the Omicron variant is fine, are the mutations that lead to immune escape. Okay, so what that suggests is, like, Omicron escaped pre-existing immunity by making mutations at loci that, if they had occurred in the ancestral background, right, they would have prevented that strain from infecting people. So like, somehow the Wuhan strain must have been evolving and getting these kind of, I don't, what is the word, like, I'm blanking on the word, but a word but the mutations that make the immune escape possible in the future. Okay, these mutations, if they happen in the Delta variant, I'm not showing this, but if you make them in the Delta variant, okay, they're also destroy ACE2 binding. Okay, so it's not that the immune escape we saw in Omicron could not have, those mutations could not have occurred in the Delta that was circulating at the time. It could occur, and in fact, you did see occasional strains that had those, but they presumably interfered with the ACE2 binding and therefore couldn't spread. Okay, here's a little bit more detail of it. Okay, these are 5 of the immune escape mutations. Okay, this is the binding to ACE2 without any of those, and this is the binding with one of them. So you can see each of them makes things a bit worse, and if you have all 5 of them, it's really bad. If you add this compensatory mutation, getting all of them is not as bad. If you add this compensatory mutation, getting all of them, well, it's almost as bad, but maybe not quite. But if you add both of these compensatory mutations together, now you can tolerate these escape mutations just fine. Skip that. We also discovered another mutation, this Y505H. This is another mutation not involved in immune escape that has exactly the same pattern. It's really bad if it happens in the ancestral background, okay? But if you add these other 2 mutations, it's no longer bad, okay? We don't have any idea what this thing is doing or why, we got it or what, but it suggests it was something important because it's something that, interferes with ACE2 binding, but you've compensated for it. So like maybe it's doing something. Okay. So again, just to sort of state the sort of general implication here, the idea is that like at that time, Delta was actively proliferating in sequences of acute infections all over the world. And all these escape mutations were happening all the time, and mutation rate's pretty high. But they couldn't go anywhere because they would interfere with ACE2 binding. So you didn't get immune escape, okay? But probably what was happening is there was some deer, or more likely some poor immunocompromised schmuck, okay, that had been infected with the original Wuhan strain for 1.5 years, okay? And in that immunocompromised person, okay, by definition, there's like not much immune pressure. The selection is relaxed, okay? And then you can get these sort of combinations of things which you couldn't get in the context of a chain of acute infections where the selection pressure on all of these phenotypes was simultaneously strong, okay? So that's kind of the idea of how this sort of thing might happen. And it sort of highlights like the importance of like, these kind of rare events driving this kind of emergence, okay, in all these, by relaxing selection on things and allowing you to cross like valleys, okay, which otherwise are preventing the evolution of these kinds of things. Okay, yeah, that's what I was going to say here. How long, when should I shut up?
Got maybe 3 or some minutes, but you can push it for a couple of minutes.
Okay, I'll try to be quick. Okay. We did the same thing with the same library. I've talked about these 5 mutations as being immune escape mutations. Why do I say that? Well, I say that because, a lot of immunologists worked on them and identified them as such. But we also did the same thing. This is that RBD again. In color are like 4 different epitopes that are like the 4 primary parts of the RBD that your immune system attacks, okay? And these are like representative antibodies, like commercially available antibodies like Regeneron, Lilly, et cetera, that bind those epitopes. So we like ordered those and we like took our library and we looked at how the immune escape to each of these things evolved. And the bottom line is there's like a whole other story, okay, which I'm going to skip. But basically, unlike the evolution of the binding to ACE2, the host cell receptor, this is pretty simple. Like most of these, just a couple of mutations, you get them, they each make you escape a little bit more, and you get some set of 1, 2, 3 of them, and you've escaped, okay? So the evolution of the immune escape is much simpler, but maintaining ACE2 binding, host cell binding while you do it is hard. So I'll just take the last couple of minutes to talk about like other things that we're sort of trying to do to understand all of this a bit more. So I've just told you 2 stories about like 2 antibodies and one viral protein. Obviously, Omicron has continued to evolve. Okay, this is like the old COVID. This is like the newer COVID. This is zooming in on the newer COVID. There's some recombinant events. There's like all kind of stuff going on in the world out there. Okay, and so we're trying to develop ways to make libraries of like all of these kind of antibodies. Of all these kind of antigens. And similarly, on the immune side, like we have lots and lots of data from people's blood, essentially, of like the evolution of antibody repertoires within people over time. And so we have similar kind of things within individuals and across the population on the antibody side. And what I'm trying to figure out how to do is basically making large libraries of specific protein variants out of phylogenies. So like if we have this complicated phylogeny, what I've talked about before was these schemes, you make these combinatorial libraries. So like I have, you know, like I have some strain here and some strain here, and there are 15 mutations, and I make all possible combinations. But that's just those mutations. What about these other paths involving mutations at other sites? And I can't make a combinatorial library of all of them. It's just way too big, right? Like you know, if I have 100 mutations, I can't make 2 to the 100 variants. But what I'd like to do is like make all of these strains and like probe how this evolution is happening, okay? And so we have a whole scheme of where we're trying to do this for influenza and for these COVID trees by making like thousands to tens of thousands of specific variants out of the trees, okay? And then being able to like track how each of these variants binds to different antibodies. And then similarly, we're making antibody trees and trying to track that. One of the coolest ones I was just talking about last week is we have some from some collaborators and from publicly available data. You all know about the bird flu, H5, right? So H5, bird flu, there are about 25,000 sequences available from the past 5 years. Most of them are from birds, obviously. There have been big outbreaks in cattle, okay? And there's also been from cattle and from birds, infections in humans, which have not made to outbreaks, but they're infections in humans. So we're like getting all these sequences and trying to make them and then ask how they bind to the bird and the cattle and the human host cell receptors, right? To sort of see like how this evolution and what are the barriers in jumping to humans. And similarly, like we're looking at this evolution here and here and trying to understand by doing this, both on the viral side and on the immune side, like how these interactions are constantly sort of running up against each other. There's a whole technical way of like how you do this, okay? Which I'm happy to talk about with anybody that wants to do it, that's interested in talking about the technical details. But basically, it's like you generate big libraries of all possible things, but then rather than try to just assemble them randomly, you basically do a kind of a scheme to get them to assemble correctly into the specific variants you want to make. And yeah, then we're going to measure neutralization and binding. All right. And I'm just going to stop there because I want to leave time if people have questions. But I want to make sure to acknowledge folks that did all this work. So the influenza work that I talked about was primarily done by Angela Phillips, who's now, she was a postdoc at the time. She just started a lab at UCSF with Thomas, Ivana, Catherine, Milo, collaborations with Thierry and Alex. And then the COVID stuff is being done, Alif, Alex, also Angela and Thomas, Jesse, Florian, and Michael are great collaborators on that. And Tatiana and Joao are new postdocs in my lab who are like doing the stuff I was, you know, talking about for the future. I'll stop there.